Thank you. Yeah, thanks, of course, to the organizers for inviting me. Arno, thanks so much for having us here. It's been a very interesting conference for me. I haven't understood fully most of the other talks, some very well, others maybe not so much. So our lab is mainly focused on, of course, cell wall synthesis, but we're taking a very much of a cell biology, molecular biology based approach to this. So in the first talk, I'm going to talk more about cell biology, so a lot of microscopy. So thank you to Mikael for introducing all of these microscopy techniques, because it makes my talk a little bit easier, I guess, as well. Really, what we're going to talk about in this talk are these two things, basically. So we're going to, I'm going to give you examples for cell wall synthesis in growing seedlings. So basically like how seedlings direct their growth. And in the second part, we're going to talk a little bit about wood formation. Both of these systems are really like cell walls are central to the development of those. So cell walls, you probably think about this, right? When you hear cell walls. So it's kind of like the walls that are surrounding us in this conference room as well. You can think about the walls as the cell walls, right? And we have the cell in the middle, right? And so with this aspect, it might be not so difficult to also put this in a pathogenic context, as we heard previous talks. Of course, the cell walls are there to defend the cells against, for example, biotic and abiotic stresses. And of course, the cell in this conference room would need to grow at some point as well, right? I mean, we know that cells are expanding. And if the walls would look like that, of course, this would be a disaster, right? So the walls would move, this roof will fall in, and probably all perish, right? So cell walls needs to be able to expand. They need to be dynamic. And this may be illustrated over here as well. So there you see a cell, basically, and the cell walls are dynamically put down around the cell, right? And you can perhaps envision this as the cell walls here mainly being laid down transversely around the cells. And of course, now when the cell expands, it has to expand in the longitudinal direction because the walls are preventing expansion in the other direction. So this is also giving shape to the cells and of course also to the plant. Right? And actually, my previous career was actually in construction engineering. It's far way back. It's probably telling you how old I actually am, right? But I think there's a nice connection between actually the walls that you see to the left-hand side and the cell walls that we're currently trying to investigate. So cell walls are not only important for plants, and they're of course also quite important for us. So for example, a lot of you are working on cotton, and cell walls, I mean, cotton is virtually just cellulose, right? And that's a cell wall component. It's also important for the woods that we are having in, well, basically where I'm standing right now, yeah. And of course also for fuels. We had this like second generation biofuel hype for a while, but cellulose was going to be degraded into ethanol and then fuel cars and so forth. It's actually also important for food and feed. For food, we heard before, like the fibers for the beta glucans, for example, in terms of actually your digestive tract. But of course, most of our cows, pigs, and so forth are actually eating plants and therefore cell wall material. And finally, it's actually our major climate change mitigating resource, because most of the carbon dioxide that is fixed through photosynthesis in plants are then being deposited into cell walls. So if we can just make the carbon dioxide stay in the walls, then of course that would be very beneficial. And so maybe the last aspect of why I think it's important to study cell walls is that these polymers make up the most abundant material on Earth. So maybe we can go into like how these cell wall polymers are made. So most of them are actually made inside of the cell. Let's see if this is working. Inside of the Golgi in here. So here you would have substrates, so nucleotide sugars, being transported into the Golgi lumen. And then basically these other enzymes in here, transferases, are stitching these sugar molecules together to form sugar polymers. And those sugar polymers are then transported from the Golgi to the cell surface, where they're kind of baked into this growing cell wall. There are, of course, also exceptions to this rule. Actually, the thing that we're mainly studying in my lab is actually occurring directly at the cell surface. So you can see here that you have these cellulose synthase proteins that are making cellulose directly at the surface. So plant cells are investing quite a lot of energy into making these cell walls. So it's estimated that 5 to 10% of the typical proteome in a living cell is dedicated to making these, changing them, or breaking them down. And so if we now skip out to the cell wall, this is typically what you would look at, right? So you have this kind of almost like a jungle of sugar polymers that are now dynamically interacting with each other. And here again, you can see these cellulose synthases that are making cellulose fibers. So in our lab, or in my lab, we're interested in a range of different questions that are related to cell wall structures. So we're interested in what the thousands of proteins that are associated with cell wall production actually do. We're interested in how these proteins and the corresponding genes are regulated. We're also interested in how they engage with each other and where this is happening. We're interested in how cell walls can communicate to the cell. Now, it has to basically tell the cell to make more or less cell wall polymers. And we're also interested in how cell walls really look like and how this relates to how plants grow. And finally, we try to put this in context of development, environment, and also evolution. And today's talk, we're mainly going to focus on this, so basically the development of different organs and so forth, and how cell walls are contributing to that. So to understand that, we first need to look a little bit more at how cellulose look like. So it's a quite simple polymer. Actually, it's just like glucose units that are stitched together in this like beta-1,4 linkages. And then they actually align with each other to form hydrogen bonds, and it makes these like very strong microfibrils or macrofibrils. And you can kind of see this as biological steel wire, because they actually have a tensile strength that is equal to steel, more or less. So you can now imagine if you wrap these cellulose fibers around the cell, of course, that is going to provide basically the stiffness or the strength of the cell wall, and therefore also like regulating the morphology of cells. So if you have this stiff cellulose microfibril coming out up here, and this cellulose microfibril is now baked into the wall. So basically it's immobilized. It cannot move anymore. What the heck happens when you make more cellulose here, right? I mean, if this guy, or like the cellulose microfibril, cannot move, what do you think will happen when you're making more cellulose here? Any bids from the audience? Do you think it will just be like that these glucan chains will like extend up like that and then going down, or like what do you think will happen? So it's probably most likely that the whole complex will move in the plasma membrane forward, right, while it's making this. And actually, through some of the tools that Michael was showing in the previous talk, we can actually visualize this directly in a plant cell. So this was done actually by the PhD student next to the lab, me in the lab in Carnegie. So I overlapped actually with Sue at Carnegie at the time. But he was making this construct. So basically, he fused one of the CSA genes, so cellulose synthase gene, with the fluorescent protein. And then they put that into Arabidopsis. And here you're looking at the basically growing, well, as a hypocotyl, right? And looking at the single cell from that hypocotyl. And I hope you can appreciate that you can now follow the cellulose synthase complex in a living seedling while making cellulose, right? And this is pretty neat, I think. And we can, of course, now begin to understand a little bit more about how this process works, right? So, for example, you can calculate the speed, right? So it's moving with 150 to 500 nanometers per minute. And you can translate that to amount of glucose units that are incorporated per time unit into these cellulose microfibers. Another interesting observation was some of you probably noticed that most of these dots are moving in this direction, right? Not in this direction. So there has to be something inside of the cell or in the cell wall that is steering where these cellulose fibers are being deposited, right? And so old electron microscopy imaging has shown, or had shown, that cellulose microfibril that you see here in orange are typically laid down in parallel to internal structures that are called microtubules. So they are a major component of the cytoskeleton. And actually what you can do now is just cross our fluorescent line with the CSAs, as you see down here, with a microtubule marker. And I hope you can appreciate that these cellulose synthases are moving on top of these microtubules when they're producing cellulose, right? So this was an elegant way of showing basically the thing that had been shown with electron microscopy imaging before. So in a way, you can now visualize this process in your mind, hopefully, like it's this kind of old steam engine train, where the train is basically the cellulose synthesis. The smoke could be like cellulose, right? And then you have these rails that are working as the microtubules. So basically, if you change these rails, you will presumably change also the direction of how cellulose is being produced. All right, so basically using microscopy, we've actually learned a lot about how these cellulose synthases are regulated during this process. Right? So by basically using a trick where you bleach a certain region of the cell and look at how fast cellulose synthases are repopulating the area, we can calculate, for example, how long the cellulose synthases are active with the cell surface. We can calculate how long the cellulose fibers are. And finally, we can count, of course, the number of CSAs per cell. And so in a cell, we typically have about 2,000 active CSAs in these growing cells that are basically making up these seedlings in this case, right? Okay, so I hope I've shown you here that we can really measure and evaluate cellulose synthesis in real time in a living plant, right? Okay, so now we're moving from these primary cell walls that are basically supporting the direction of seedling growth to secondary cell walls, so basically wood-producing cells. And so there's some differences between these primary cell walls and secondary cell walls. And so primary walls that are mainly made up of these sugar-based polymers, right? They surround each and every plant cell, and they're very dynamic. Secondary cell walls, on the other hand, they also contain these sugar-based polymers, but also something called phenolics or lignin. And they typically are made around or in woody tissues, and they're really stiff and strong. They're kind of the concrete of cell walls. And so you can appreciate that perhaps here, if you look inside of a stem of Arabidopsis in this case, you can here see we have stained just these secondary cell wall producing cells. So you have the xylem that transports water, and then you have these fiber cells that give stature to plants. And of course, these secondary cell walls were necessary for colonization of land by plants, right? Because they now allow water to be transported to the aerial parts of plants. But they're also key, of course, to biomass of the plant. So they're really important for a lot of different aspects for industry as well. Right. So just to give you an example here, actually, if you now look at water, it's transported through, in this case, a tree, right, up to the canopy. Actually, most of the water is evaporated. It's about 95% of the water that is transported up to the canopy is actually like evaporated out through stomata, right? But then you have this water that is now conducted through this system, and you can probably appreciate that as just cells that are aligned with each other and are dead cells actually that then conduct this water up to the aerial parts of the plant. So this, of course, needs to be quite strong, because you can imagine that this is almost like a straw in your soda, right? You suck on that straw, and you need to have a sturdy straw to actually support that, right? So if you don't have that, actually my postdoc, he was from East Germany, and he compared these straws that they got when they were drinking Cuba Cola, I think in East Germany at the time. They were not very sturdy. So when you sucked too hard, actually this straw is just like buckled in, right? So they basically, this pressure, inverse pressure that you're generating is basically collapsed the straw. So you wouldn't get like any Cuba Cola in that case in your mouth, right? And this is actually the case as well. So if you're mutating some essential cell wall components that are associated with secondary wall synthesis, you can here see that you're getting these collapsed cells as well, right? And this is due to this like negative pressure that is generated. So to support this, right, both to give the strength, but also to transport water, you need to have like certain structures of these secondary cell walls. And you can see that they have different structures. You have this like, let's get banded or spiraling patterns, or also pitted, or basically like holes in the secondary cell walls. And so in our case, we were really interested in trying to visualize this process in real time. So basically visualizing wood formation in real time. And this is quite challenging, because most of these wood forming tissues or xylem cells or xylem vessels, they're buried inside plant organs, right? So you have multiple cell files that you need to penetrate to basically get the imaging going. So in our case, we actually took a shortcut to this process. Basically what we did was to tell the plant to make secondary walls, or this woody cell walls, in all cells. And we did this by introducing this construct here. So basically this is VND7. It's a master regulator for secondary cell wall synthesis. So basically if you turn on that transcription factor, all of the cells will turn into wood, in a sense, right? And so how this is working is that typically you express this VND7 here, right? But under normal conditions, so without this inducing factor, it's tethered to heat shock protein in the cytosol. And so then it cannot go into the nucleus, and it cannot activate this process. Right? But of course, if you add this inducing factor, we can now like basically get this transcription factor into the nucleus. And therefore, you can now actually turn on secondary cell walls in all cells in a seedling. And just to zoom in, you can hopefully appreciate here that you have these bands that are now surrounding all of these cells in a seedling. And so basically now we have to go back to the first part of this talk, because I guess now we're actually moving away from making just these primary walls that are kind of evenly distributed across the cell to these banded patterns, right? And if you now remember the first part, then the cell wall, I mean, in this case, cellulose, they're basically guided by these rails, right? So our prediction here would be that the microtubule organization, or these rails, would change from this more random organization, or like actually like evenly distributed organization, to these bands. So what we did was just to introduce a microtubule marker in this VND7 line, and I hope you can appreciate here that over time after this induction, you can see this evenly distributed microtubule array that is now changing to this banded pattern, right? And now we can, of course, follow this in real time in a cell. So our movie is actually around 10 hours long, but we've cut it down here in interest of the coffee break. But you can hopefully appreciate that we can now change this microtubule array to this banded pattern instead. And actually, we can now take a lot of different tricks in terms of bioimaging analysis tricks. And basically what we're doing here is, in this movie that you see over here, we take two next door images to each other and we overlay them on top of each other. And then we subtract those images from each other. And that's basically what you're now seeing, hopefully, in these channels. So these are growing ends of microtubules, and these are shrinking ends. And you can hopefully visualize or see that over here as well. And so we now measured many different aspects of how the microtubules were behaving. And the thing here was to try to recapitulate this in a model. And so that model, we basically made this in a computer, right? Where you have the cylinder that we populated with sticks. And those sticks are behaving like the microtubules. And I hope you can appreciate that over time, we can begin to recapitulate how these patterns are emerging. Takes a little bit of time, I'm sorry. So you can now see that we're beginning to form these bands also in the model, right? And so we think that this is a really neat way of combining basically biology with modeling, right? And so basically by changing or tweaking our model, we can now begin to see what do we need to readjust for this microtubule behavior to make different types of patterns. And of course, this has some biological relevance as well, because we have all these different patterns of secondary cell walls that we now potentially can explore. Right. So we think that this is giving us some form of possibility to tweak these cell wall patterns, and therefore also tweak perhaps how these secondary cell walls are made. So this part of the talk was basically to understand how wood is patterned and potentially also give us a way into making new type of patterns in secondary cell walls. So you might wonder, okay, you have only looked at these microtubules, right? So what's happening with this cellulose synthesis during this time? And actually, this is a really interesting question, because we have one protein complex shown up here that is involved in making primary cell walls. But that is shifted out to another protein complex that is now making cellulose during secondary cell wall synthesis. And so our thinking here was that if we now have a marker for this primary wall complex up here, together with another marker for the secondary cell wall in different colors, and we cross that into this inducible line, we can now look at basically the transition between making a primary wall to a secondary wall in real time. And this actually turned out really nice. These are just stills from that, but anyway, I think that you get the picture. So you can see the primary wall is made, of course, before we are inducing. And then you can see that we slowly begin to form these bands, right? And there you can also see that you're slowly now, the primary walls are disappearing, right? So you're not making any more primary cell walls. On the other hand, you can see that you're inducing now these secondary cell walls, and that you're again having these patterns. What's really neat is that actually you have an overlap between the primary wall synthesis and secondary cell wall synthesis. So now we can begin to look at okay, are these complexes actually moving in the same way, right? And so we quantified this, and we found out that actually the secondary cell wall complexes move twice as fast as the primary one. So that means that they're actually spitting out cellulose twice as fast during the secondary cell wall as the primary cell wall. So in a way, you can kind of imagine this as this passing lanes on a highway, right? Where you would have the primary wall assays probably moving in the slow file, and the faster secondary cell wall using complexes moving in the fast file. All right. So these are kind of ways to visualize cellulose synthesis and pattern formation in woody cell in real time. So I'm going to talk a little bit more about microscopy as well. So maybe a little bit like along the line with what Mikael was doing, but we're actually not using so much these off-the-shelf company-based systems. So we're actually trying to modify the systems to suit our purposes in the lab. And so one system we built together with 3i, you can see here probably. So it's a regular spinning disc confocal microscope that Mikael was also talking about, but you can probably see that we, instead of having this horizontal stage, we actually now have a vertical stage instead. So in this case, all you do is like unscrew your objective down here. You put on this objective inverter and then screw on your objective over here. And now you can image basically on this vertical stage. And for plant biology, kind of makes sense, right? I mean, plants are typically not growing in this direction. They tend to grow vertically. So you can now begin to image things in real time over longer periods of so many labs now have these like vertical stages as well. They don't need to be that expensive, actually. It's this was a really cheap solution to the system. But the thing is, like now, most of the imaging that we do on media plates, and we all know that plants do not grow in media out in nature, right? So in the lab, we're trying to get around this problem and actually like make small incubation chambers where we can grow plants directly in soil and to image them in soil, right? So we make small rhizotrones, if you want. So this is just like things that we've developed in the lab. So here you have an incubation chamber. You can see that there's soil, right? And you can now, for example, now follow this root throughout the soil the whole way, right? And you can now, of course, zoom in on any region of this root that you're interested in. You can, of course, also now begin to follow root growth in real time. And that's what we're doing here. For example, you can now see that you can really follow these roots nicely with a plasma membrane marker in soil, right? And so in the lab, we're quite interested in compacted soil conditions, right? So it's a major agricultural problem. So basically in the next one, you can see now a root growing in compacted soil. And you can hopefully see that the length of the cells are actually much shorter and the root is more swollen. And that's a typical response you're getting from compacted soil. And so of course we can take this further. What we've done now is basically combine different soil types in one chamber. So now you have compacted soil down here and non-compacted in the top. And we can begin to look at now how the root is behaving in real time when they're basically like hitting this compacted soil, right? And so this is just a postdoc here, and here's just like one root that we're following through this like compacted condition. So as of course, we have only tapped the surface of what we can do with this system, I would say, right? So you, of course, can now begin to look at like how are the different cells behaving when they're hitting, for example, this compacted soil? How do different marker lines for like hormones and for different signaling components also behave during this process? And this is really important for cell biologists to understand, to actually understand how this system is reacting to these conditions. Right. And so we think that this can be used for a lot of different purposes. I mean, in terms of root microbe interactions, for example, different types of abiotic stress responses, and of course, understanding signaling in roots in real soil, or at least like as close as we're getting. We can perhaps try to like dig down a confocal microscope in soil, right? But I think this is probably the closest we're going to get. Right. So back to this confocal microscope that was shown before as well, right? So I know that this is pretty pricey. I mean, I think an investment for a new confocal microscope is pretty expensive. I think we paid 10 billion Uzbeki som, I think, for our system. It sounds like an insane amount of money to me. It's about 5 million Danish crowns. So that's more reasonable, I would say. But you don't need to buy these systems, right? You can build microscopes yourself as well. You just need to have the right people around. So in my lab, we have 3 physicists that are basically building microscopes themselves as well. And so this is one system, and it costs actually 0 crowns or 0 sols, because we could use material from other microscopes and just take those out and reassemble them into a new system. And so this system is quite unique because most of the stuff that we are doing in cell biology in plants is based around fluorescence. Right? So you use these fluorescent proteins, you use different dyes, or you use antibodies, right? But you and me, we don't use fluorescence, right? I mean, I know that that's Barno. I know that that's an empty chair. And we're using light interactions with surfaces. So it's light scattering and refraction and reflection, right? And we interpret this in a really, really good way. And so this microscope is basically set up to combine fluorescence microscopy in this light scattering microscopy. So basically, if you follow here, you have like basically light coming in, you have your sample up here, and then we basically divide the signal into either the fluorescence, whatever wavelength we're interested in, or actually detecting the scattering light. And so this is just a surface of a leaf, and this is a single cell here, basically these epidermal pavement cells of Arabidopsis. And this is with this light scattering channel. And I hope you can appreciate that there's a lot of movement of different stuff in here, right? And this is basically capturing all organelles and bigger vesicles. So the resolution is about 150 nanometers in real time in these cells, right? This is kind of cool because there's no bleaching now, because we have no fluorescence. So you can image this for days if you want, right? Problem here is we don't know what the heck this is or this is, right? So that's why we need this fluorescent channel to be able to pinpoint down what are those different organelles or vesicles, right? But of course, over time, we will be able to track these, and I hope you can appreciate that we're tracking here now different organelles and vesicles in root hairs in this case, right? But we can learn from this. We have basically AI and machine learning tools that we now can begin to tell, okay, this is behaving in this way. And over time, we believe that we can actually use this to say, without the fluorescent channel, this is this and this is that, right? And we think that this is opening up like a completely new field in terms of doing cell biology in ops, right? We all know how difficult it is to actually do cell biology in any types of crops. And I think this might be a way around this. So what can this be used for? So we can image, of course, vesicle organelles in crop plants and other organisms. We think that we can use this, actually, we are pretty sure that we can use this. We're doing it currently for infection, for example, and plant pathogen in barley, and of course, any other crops as well. And we think that we also can use this to couple to the soil chambers that I was just showing you as well, to actually get like a better view of non-fluorescing, I mean, basically non-cell biology tractable systems, to actually look at those in real time as well. Finally, okay, yeah, actually, so these are some of the people. We have this experimental microscopy lab. So we're actually building, I mean, not only this microscope, we're actually building microscopes where you can look at mechanical stresses in tissues and cells, super resolution microscopy systems as well. Most of those cost a fraction of what a normal microscope would cost that you buy from a company. The last thing, I didn't really know what people might be interested in this context, but we're also using something called optical tweezers, where you capture you can capture just like a vesicle or compartment or whatever using a pretty high powered laser. And what you can do is, well, you can do a lot of things. You can actually measure forces between proteins with the system. But in this case, we have used it to fuse protoplast and vesicles. So you can basically now introduce new material into a protoplast by just fusing the vesicle to it, right? And so here is just like actually in this case, we're fusing 2 protoplasts together, which I don't know what the purpose of that actually will be, right? But we can do that. But of course, you can now begin to introduce new proteins, new type of lipid environments into protoplast and see how your proteins of interest are reacting to that. So with that, I of course want to thank the lab that you see here in the back. Actually, most of the work that I was showing today was done by Renee and Arun. Arun is now a group leader at the Max Planck Institute, and Renee has an emenuta at the University of Potsdam, so they were fairly successful afterwards. And then we have collaborators for these specific projects, and of course also funding organizations that made this work possible. So thank you very much, and I'm happy to take any questions.
Yeah. Thanks so much, Staffan, for the wonderful talk. So yeah, we can see that this microscopy tool is really powerful, that they can show us really wonderful view things happening in this cell, so which we cannot see by our eyes, right? Naked eyes. Yeah, then now time to discuss.
It's the microscopy mafia.
Right. Bad questions. No, thanks a lot for a really impressive talk. And I really appreciate that you mentioned that you don't always have to buy the fancy stuff. If you do it self-made, anyway, it's better because it's adjusted exactly what you want. Two questions. One is about the little rhizotrope. Because I mean, nevertheless, you have to have the surface where you can image.
Of course.
So that makes a difference also.
Of course makes a difference.
Because otherwise Blue Root would take another way, probably.
Yeah, I would still argue that it's better system than the media, basically.
Exactly, yeah. And one question about the optical tweezers. Because there it's similar. You use it only for protoplasts?
Currently, we're using it for protoplasts and different sized vesicles, yeah.
Because I think also for the cell, if it has a cell wall or not, makes also a big difference.
Yeah, of course. In this case, we're interested in introducing, for example, a new sterile environment and to see whether proteins that we are interested in actually goes to that environment or not. But you're right. Of course, any type of experiments that you're doing on protoplast have to take that very careful. It's not a plant.
Yep.
Right. Thank you.
Thank you for a talk. I'm also want to ask for the protoplast because we're working on the system, and we had quite a problem to isolate protoplasts from woody plants. So actually, we are struggling a lot isolating protoplasts from tea, Camellia sinensis, and it can probably be related to the cell wall structure. And this is actually what would you suggest to do to make it simple? Because we just take at the end we take the younger leaves ever possible, and we have some yield. But as you showed here, it is possible somehow to increase the woody cell type formation. Are there any ways to decrease them, or somehow treat woody plants in tissue culture to make protoplasts more available?
It's a good question. Yeah. We are struggling ourselves actually also to make protoplasts from certain species. We're working a lot with red macroalgae now, and making protoplasts for those has not been trivial. So it's a really good question. In terms of, I guess you can use tissue cultures, right, to try to get more cells that don't have the secondary cell walls, and maybe that will help you to get more protoplasts out from it as well. I don't know.
It's other way around. So actually, the colors. Yeah.
I'm sorry. I'm not sure that I will be the best person to help you there. Sorry.
Maybe I missed. So you overexpress this factor which made secondary wall in all tissues, and maybe I missed this. What phenotype did you get?
Oh, it's a terrible end. The seedlings die, because they're all dead cells, right? So the whole seedling just turns yellow and dies. So in terms of phenotype, it's a rather boring phenotype, I would say. Yeah.
Did you try to knock out this factor?
If you knock it out, or You cannot knock it out. I mean, I think the seedlings will die very, very early because they're unable to transport water and they're unable to stand up. So that would also not be a good end, I think. So.
May I ask a quick question about the same? So you showed that the primary cell wall stops producing, secondary starts producing. Does it mean that after this moment the cell cannot move anymore, grow, or bend to the side?
Yeah, so there is some flexibility left in it. So when you make these like they're called proto-xylem, so it's basically the first forming xylem, then these banded patterns actually allows the cell to expand a little bit, right? But when you make these pitted patterns or the holes, then it's impossible to expand anymore. You can probably think about that as well logically, that if you have just like holes, then expanding that cell wall will be extremely difficult, right? So typically, like the protoxylem formation, you have some expansion, but the cells ultimately die anyway, right? So there's a programmed cell death program that is kicked in.
So it means the secondary cell wall formation is somehow related to the programmed cell death.
To what?
To the programmed cell death afterwards.
That is the end of that process, yes.
Thank you.
Thank you so much for your interesting information. I would like to know the difference molecular density of synthesized cell walls because, as you know, salination is one issue in global change, climate change. And this reason, plants lose forcibly water content because of osmotic pressure in saline condition. In this reason, I would like to know how synthesized cell wall reduce pressure, osmotic pressure in saline conditions. Thanks so much.
So it's I mean, in terms of osmotic pressure from the inside, you mean in that case, or?
Yeah, inside.
Right. I mean, so the vacuole is taking up water that is pressing against the cell, and that actually leads to typically more cell wall synthesis and expansion of the walls. So that's basically like how plant cells grow, right? So I'm not sure if that's answering your question or not, though.
Thanks.
Okay.
Sorular var mı? Bu mesela hazır coffee break bizde. Thanks so much, Stefan, for the nice talk. Now we have coffee break, and after the coffee break, we have 2 more presentations. Then we close the workshop today. So see you later.